Hey, it's Mr. Mazurkewitz, and in this video we're going to be taking a look at the process of cellular respiration. Now, cellular respiration is the process by which organisms are going to take the energy from food and transfer that into molecules of ATP, a usable form of energy. And I wanted to start by asking you a couple of questions. So I have a few pictures of different organisms from different kingdoms here. So we have a plant, we have a mushroom, which is fungi, we have an animal, we have some bacteria. And if I were to ask you which one of these organisms does photosynthesis, what would you say? Now, if you understand photosynthesis, which I'm hoping many of you do, you would say, well, definitely the plants, they're photosynthetic, they're going to make food. If you really get photosynthesis, you'll remember that some bacteria can, so maybe the bacteria does it. You know, hopefully, that animals and fungi are heterotrophs, so they're actually going to eat their food. So you'd be correct. But what about if I asked you which one of these organisms does cellular respiration? Sometimes people think, oh, well, just animals. They're the ones that breathe, so they do it. Or maybe the animal and the fungi because they're both heterotrophs. But the correct answer is actually going to be all organisms. All four of these, and literally just about all organisms on Earth, are going to do some form of cell respiration because, again, this is how you're going to take energy from food that you either eat or make and put it into ATP. Organisms cannot survive without having a usable form of energy in the form of ATP. So in this video, we're going to cover a couple of things. First, you should understand, well, what are the reactants and products of cellular respiration? You should know the formula, know which ones do we use to begin with, and which ones are we creating by the end. And you should also have a pretty good idea about how the basic process works or how it occurs. So what are the different steps involved? By the end of the video, if you can do that, you know you're in great shape. So before we can get into the whole process of cellular respiration, again, it's important to understand, well, what is the whole purpose? The purpose of cell respiration is to take the energy stored in food. The most basic food molecule we can talk about is glucose. There's a lot of energy stored in the bonds of that glucose molecule, and we want to take it and put it into molecules of adenosine triphosphate, or ATP. ATP is going to be a portable, usable form of energy that all cells on this planet are going to use. Whether you're a bacteria, plant, animal, fungi, we all need to use ATP to do things like grow, reproduce, move, just about everything you can think of. So the formula for cellular respiration shown here, you need to understand this. You should know the reactants on the left side. You should know the products on the right side. Now, it's really nothing new. It's just a matter of understanding photosynthesis. If you know that one, just reverse it. Remember that in photosynthesis, plants are going to use carbon dioxide, water, and light energy. They're going to use those to create oxygen and glucose. Well, in cell respiration, organisms are going to take that same glucose and oxygen, and we're going to use that to create carbon dioxide, water, and energy, but this time that energy is in the form of ATP. So remember that all organisms are doing this. And again, just looking up here, we have autotrophs. If you create your food, or if you're a heterotroph, you eat your food, you're going to use things like organic compounds, so food. You're going to take in oxygen from your environment, and you're going to produce carbon dioxide. You're going to get rid of it. You're going to produce water. You're going to get rid of that. But the key one that we want to get here is our ATP. Now, where are we going to do this whole process? One organelle should come to mind when you hear cell respiration, and that organelle is the mighty mitochondria. Now, even though mitochondria is not going to be the site of all steps of cell respiration, it is where the bulk of it will take place. And before we get started, I just wanted to go through some parts of the mitochondria. So if you recall, the mitochondria actually has two membranes. There is an outer membrane that is shown here, made out of lipids, as well as a folded up inner membrane. So that's usually how we recognize the mitochondria. It's got that double folded inner membrane. Now, inside the center, within both of those membranes, is an area here I'll refer to from time and time again. It's called the matrix. This is where a lot of chemical reactions will be taking place. And then you even have what's called the intermembrane space. That's the space between the inner and outer membrane. Now, remember that mitochondria were once bacteria. So even within the mitochondria, you'll find that it has its own DNA. It's able to divide on its own. It's got its own ribosomes. And remember that endosymbiotic theory, which states that bacteria were taken in by cells, and those are what became eventually things like the mitochondria and even chloroplasts. So let's get started with our first step of understanding how we can go from the energy in glucose to actual ATP. The first step in cellular respiration is going to be a process that we call glycolysis. And I wanted to point out that where this is taking place in the cell is actually inside the cytoplasm. We're not inside the mitochondria yet. This is going to take place right outside the mitochondria in the cytoplasm of the cell. And if we were to take a look at this word glycolysis and break it down, we've actually got two parts here. The first part is glyco, which means sugar, and lysis, which means to break apart. So literally, what glycolysis is all about is taking our molecule of glucose to start our food, and we're going to break it into two. So we're going to take a 6-carbon sugar. Remember that glucose is C6H12O6, and we're going to break it into two pyruvate molecules, each with 3 carbons. So we're taking a 6-carbon molecule, breaking it down into two 3-carbon molecules. What do we get out of this? Well, just simply by doing this process, we're actually going to get a little bit of ATP out of this. We're actually going to net 2, and the reason I say net is because it's going to cost us 2 to do it. We have to use 2 ATP. We're going to create 4 from the process, so if you take 4 that we create minus the 2 that we had to use, we're actually only netting 2 ATP. Another thing that we're going to get out of this are some of these high energy electrons, and these aren't important yet, but you will see that these high energy electrons will be used later in the process of cell respiration. So to just once again go through this process, we took glucose, we broke it down into 2 pyruvates, we got 2 ATPs and some high energy electrons. Now it's also important to note that oxygen was not required for this process, and because oxygen is not required to do this, we call glycolysis an anaerobic process. This is a term you must know. Anaerobic means oxygen is not required, and you'll see why this is important in another video when we talk about anaerobic respiration. Now that we have our pyruvates, we've created some ATP, we've got our high energy electrons, we're able to continue to stage 2 only if oxygen is present. So in the presence of oxygen, if you take in oxygen, we're able to move to step 2, which is going to take place now inside that mitochondria. So in the mitochondria, we're going to take our pyruvates, we're going to move them inside to the matrix, as well as those high energy electrons, and those 2 ATP can go be used somewhere for some sort of energy. So now we're going to move on to stage 2, which we call the Krebs cycle. And now this is going to take place inside the mitochondria. Again, we're in the matrix now, inside those 2 membranes of the mitochondria. Because the Krebs cycle can only take place if oxygen is present, we're going to have a different word for this. Now remember, glycolysis was an anaerobic process. The Krebs cycle is going to be referred to as an aerobic process. This can only happen if oxygen is present. So aerobic means oxygen present, while anaerobic means no oxygen is required. Now in the Krebs cycle, it's a whole series of steps. You're not required to know them all, but pretty much we want to know what do we get out of this whole process. So through the Krebs cycle, we're going to take those pyruvate molecules that we made from the glycolysis, and we're going to do a series of chemical reactions, and every time a pyruvate goes through this whole cycle of the Krebs cycle, what we get out of it is a molecule of ATP. Now remember, we have 2 pyruvates, so each one's going to go in, and as a result, we're going to get 2 more ATP out of this. So just to tally up our count, remember we got 2 ATP from glycolysis, 2 more from the Krebs cycle, so now we're up to 4 ATP total. Now when we're breaking down these pyruvates, remember those had carbons. What happened to the carbon that made up those pyruvates? Well, we no longer need them, so once we break it down, we're actually going to get rid of some of that carbon in the form of carbon dioxide. So every time that you exhale, you get rid of carbon dioxide, and where did that carbon come from? It was really from that Krebs cycle. So in addition to the ATP we've created, we've gotten rid of some carbon dioxide. What else do we get out of the Krebs cycle? We get some more of those high energy electrons, which again will be used at the last step along with the ones that we got from glycolysis. So again, to recap the Krebs cycle, we took pyruvate, we got 2 ATP, we got rid of some carbon dioxide, and we've gotten more of these high energy electrons. Now we're going to move on to the very last step of cell respiration, which is called the electron transport chain. Now again, this also only happens if oxygen is required or if oxygen is around, so it is going to be an aerobic process. Now the electron transport chain, what we're going to use is the electrons, the ones we got from glycolysis and the ones that we got from the Krebs cycle. And remember, those are high energy. We're going to use that energy to kind of drive this whole chain reaction. That's why we call it the electron transport chain. Now it looks a lot more confusing than it really is, so try and follow along. Here we are in the inner mitochondrial membrane, or that inner folded up membrane of the mitochondria. And at this point, what we're going to do is use these high energy electrons, shown by these stars here, and they're going to move along from protein to protein. And as they do that, they're going to use some of their energy to pump, using these protein pumps shown in red, we're going to pump positively charged hydrogen ions to one side. So think of this as like a dam. If you were to take a dam and put all water on one side, you're building up a lot of potential energy. That water is going to want to flow down to the other side. Well, we're doing the same thing here. We're using the electrons to pump hydrogen, positively charged hydrogen ions to one side so that we're building up this potential energy, because remember, things want to go from high to low concentration. Well, what's going to happen once we open up that dam, or we open up the floodgates? Well, over here we have another protein that we call ATP synthase. But pretty much what this is this is going to use the energy as these hydrogen ions flow back through. When we open up that dam, that's going to provide this guy with a little bit of energy to create ATP. We're going to take ADPs, we're going to throw a phosphate onto it, and what do you get when you take an ADP and add another phosphate? You get your ATP molecules. Well, how many ATPs are we going to create through this process? Well, every time a hydrogen ion does it, we get ATP. We're going to create a total of 34 ATPs through this process. So that's a lot of ATP. How did we do that? We just moved all these hydrogen ions to one side and then let them pass through, and we use that movement, that energy, in order to generate some ATP molecules. Now, one last thing we have to mention is, well, what happened to those electrons? Remember that they pass through, we use their energy, but once they get to the end over here, what is their fate? What happens to them? Well, this is where oxygen is going to come into place. Oxygen is what we call the final electron acceptor. It is really good at accepting and taking those electrons. So what oxygen will do when you breathe in oxygen, it's going to go into your cells, grab those electrons, also grab two hydrogens. So what happens when you get oxygen combined with two hydrogens? You get a water molecule. So when you breathe out, not only are you breathing out carbon dioxide from the Krebs cycle, but you're also breathing out the water that you made during the electron transport chain. And oxygen is crucial because, again, without oxygen, you have nothing there to take those electrons. That's going to stop this whole process from happening, and you won't be able to create your ATP. So why is it that you suffocate and die if you can't breathe, you can't get oxygen? It's literally because there's nothing there to accept those electrons. You can't do the electron transport chain. You're not producing enough ATP, and you can't survive without it. So with all this said and done, let's just recap the overall products of cell respiration, go through the whole process again. Remember that in the beginning, we started with a molecule of glucose, C6H12O6, and we use that through the process of glycolysis. It's an anaerobic process, and from that, we got 2 ATP. So we got 2 from glycolysis, and remember, we created some pyruvate and some high-energy electrons. We then took those pyruvates and we moved on to the Krebs cycle inside the mitochondria, an aerobic process that requires oxygen, and we got another 2 ATP, so we're up to 4 now. Remember that we also got rid of some carbon dioxide at this point, so we're going to put that on the product side. So we breathe out the carbon dioxide from the Krebs cycle. We're then going to take those high-energy electrons from glycolysis from the Krebs cycle. We're going to move on to the electron transport chain, where we're going to create another 34 ATP. And remember that we needed oxygen for this step, so we're going to add that to our reactants. The oxygen combines with those electrons, some hydrogens, and turns into water. We're going to get rid of that, so that's going to be a product. So that brings our total ATP count to 38 ATP per molecule of glucose. For every 1 molecule of glucose that you take in, your cells are able to create roughly about 38 ATP. So here's our whole chemical equation. We have glucose plus oxygen, the things that we took in. What did we create in the end? Well, we got rid of carbon dioxide, we got rid of water, and we created 38 ATP that your cells will use for just about everything. So at this point, hopefully you're able to go through those 3 parts of cell respiration, understanding basically what they're doing, and really you should have this formula down. You should know how the reactants of cell respiration are used to create the products. If you're having a little bit of trouble, feel free to go and rewatch certain parts or ask me any questions that you might have, but thank you guys so much for watching.